Hello, everyone. On behalf of HistoIndex and Global Engage, welcome to Deciphering NASH, Fibrosis Dynamics in Cirrhotic Patients and Insights into Ballooned Hepatocytes using AI. I'm Cynthia Ann Victor from HistoIndex, and thank you for joining us today. We have for you presentations featuring artificial intelligence data on Septa, nodules, fibrosis, and ballooning. These data have demonstrated the stain-free AI Digital Pathology platform by HistoIndex as a reproducible tool in improving drug efficacy readouts in NASH clinical trials. Presented by global key opinion leaders in NASH, our webinar presentations aim to provide an in-depth understanding of how key features and subtleties of NASH can be fully quantified in an accurate and consistent manner with stain-free AI digital pathology with a second harmonic generation and two-photon excitation fluorescence technology. The collective takeaways from these presentations will benefit pharmas and biotech companies involved in therapeutic discovery and drug development for NASH. Please note that the content showcased in this webinar is for educational purposes. The presentation slide decks are the property of the presenters and should not be reproduced without consent. This is our agenda for today, and please note that the Q&As with our guest speakers will take place after the second presentation. Following the Q&As, my colleague Anthony Lai, the head of clinical affairs at HistoIndex, will share some brief information regarding the presentations, so please do stay with us. Here are some house rules before we get started. All participants will automatically be muted during this webinar. A gentle reminder that this webinar will be recorded and be made available on demand to registered attendees. Email notifications will be sent to you in 3 working days after this event. If you need to send us a message, all you need to do is to select the chat icon that's circled below. And after the chat panel appears, you may select either the moderator or the host's name from the drop-down menu as seen below, and you may enter your message in the box. Our 15-minute Q&A segment will be moderated by Anthony, and he will be taking your questions to our presenters. Therefore, please feel free to pose your questions at any time throughout the presentations. To send in your question, click on the Q&A button that you see on the bottom panel of your Zoom screen. Once you see the Q&A pop up on your screen, kindly include your name and the required details. We would encourage you to leave your details so that we can be in touch with you in the event that your question remains unanswered in the Q&A segment. I'm very happy to introduce our presenters for today. First, we have Dr. Mazen Noureddin, Founding Director of the Fatty Liver Program at Cedars-Sinai Medical Center, Los Angeles, California. Hello, and thank you for being with us, Mazen. How have you been?
Hi, how are you? Thank you for inviting me.
Thank you, and we're really happy to have you with us today. And yes, thank you. Let's meet our next presenter, Professor Quentin Anstey, Chair of Experimental Hepatology and Consultant Hepatologist from the Faculty of Medical Sciences at the Newcastle University, UK. Quentin is also an honorary consultant hepatologist of the liver unit at the Freeman Hospital in Newcastle. Hello, Quentin. Thank you for being here with us. How are you doing?
Very well, thanks, Cynthia. It's a great pleasure to be here, and thanks very much indeed for the invitation to participate.
Oh, it is indeed our honor and privilege to have you both on board to share your work with all our guests. So thank you both for being our presenters. So to our dear participants, I trust that you will enjoy the presentations and the key takeaways. Without further ado, let's welcome Dr. Mazen Noureddin with his presentation titled Quantitative Assessment of Septa, Nodules and Fibrosis using Stain-Free AI Digital Pathology shows excellent correlation with portal pressures and varices in NASH patients with cirrhosis. Take it away, Mazen.
Thank you very much. Share my screen. Fabulous. Can you see my screen?
Yes,
Yes.
perfect.
Great. So, good morning if you're in America. Good afternoon if you're in Europe or the Middle East. And I don't want to say good night for people in Asia yet. We will say that after the Q&A. So stay up with us. We have some exciting research and information to present to you today, me and my esteemed colleague, Professor Quentin Anstey. So I want to start with a background about cirrhosis. And let me say this, solving cirrhosis is the passionate spot in every hepatologist's heart. So all of us want to try to cure cirrhosis at one point in the future. And to do that, we need to understand cirrhosis, how to classify it, treat it, and grade it histologically very well. So I don't need to share with the audience what is cirrhosis. It's the later stages of the liver disease. When you have stage 4 fibrosis, it depends on the histological score you're using. But also, after you get to cirrhosis, data over and over the years have shown that at the beginning of cirrhosis, patients are not in trouble, but eventually they get into trouble when they develop portal hypertension. And there's plenty of research came on that topic and how to quantify portal hypertension, usually using this hepatic venous portal pressure gradient that, as you know, we measure through transjugular liver biopsy. So that term coined as clinically significant portal hypertension gradient, which is HVPG, which is hepatovenous pressure gradient more than 10. And it became a foundation of our studying of outcomes because that portal pressure correlates with outcomes. Also, before you get to the clinically significant portal hypertension, there are earlier stages of cirrhosis. And to quantify them, so far the best way has been histological methods. But the issue with the histological methods or conventional histological methods that it could be too simplified by looking into just like fibrosis changes, for instance, from stage 4 to stage 3. And I think me and my presenter colleagues, we have presented in many conferences that it's really different going from stage 2 fibrosis to stage 1, compare stage 4 to stage 3, or 4 and 3.5 even. This is not apple to apple or oranges to oranges, because cirrhosis is way more complex pathophysiology, architect, and many other things. So rather than looking at just like a line of fibrosis, there are other things to consider. For instance, when you get to cirrhosis, you get the nodules. And those nodules are not created equally. They could be small, they could be large, they could be intermediate, and each one of them has its weight on the stiffness and eventually the portal pressure. In addition, we know that what bridges between portal tracts and connect them eventually to get cirrhosis are septas, and those septas vary among patients and vary in the same patient along the cirrhosis process. And I'll show you a few examples in the next slides. So back in the 2000s, and I would like to acknowledge Professor Garcia et al. And Professor Gruzman. In addition, also Dr. Scott Friedman joined them eventually. They described these phenomena in cirrhosis, and they called for subclassification of cirrhosis. I show you on the right, for instance, different size of nodules. Look at the top, the nodules are different from the nodules in the middle, that they look mixed. They mix between small and larger, and on the bottom you have larger nodules. Also on the left, I show you the septum. And don't tell me the septum on the top that looks thin is similar to the one in the middle, like the patient in the middle has kind of like thin septum on the left side and a larger one on the side. This stiffness will be different than this stiffness. And absolutely the last one will have way more collagen, way thicker septum, and likely they will get to portal hypertension closer. And think about drugs that target fibrosis. Like these two in the bottoms will be the hardest to treat, versus the two on the top, probably they will be easier. And thus, that concept made a lot of sense that cirrhotics are not created equally. And Professor Garcia et al. Have done few studies incorporating the concept that you should not just look at this fibrosis changes, rather include the architect of cirrhosis, fibrosis area, septal thickness, as well as size and number of nodules. And she has done research on mainly hepatitis C in the 2000s era, where the number of patients were limited in that study. And she concluded an important finding, that she found in some study that nodules correlated with HVBG. At that time, they were measuring VBG as a surrogate biomarker of outcomes. And in other studies, it did not. So brilliantly, they said, this is actually likely related to the semi-quantitative techniques we're using, the variation in pathology reading, and maybe it can be much better if we don't have semi-quantitative. And this is where the concept of the research by HistoIndex and our study came. So using second harmonic generation two-photon excitation fluorescence, a machine learning algorithm, we incorporated these concepts, the fibrosis, and we took many parameters for fibrosis, septa, including many parameters of septa, and nodule, including many parameters of nodules. And we have the advantage of machine learning here that allowed us to look into many hundreds of these parameters. And what we did is we took advantage of some of the NASH cirrhosis study. I remind you, the studies by Garcia et al. Were done mainly in hepatitis C, that luckily we're getting it to be eradicated soon. We took advantage of NASH cirrhosis study, which is the vilipectin study that measured portal pressures as the primary outcome. It also included assessment of varices. So here we have a surrogate of outcomes, which is H3BG, and also an outcome, which is presence of varices or not, to compare to. And the concept is, instead of doing triangular liver biopsy and measuring pressure, can these detailed histological parameters, including fibrosis and septa and nodule, especially with using the HGG TPEF, the second harmonic generation machine learning algorithm, can tell us what the portal pressure and varices look like. And this project has been ongoing for a few years now. But a little bit quick on the technology of second harmonic generation, and now it became very well known and widespread. It is a reading of tissue's endogenous biomarkers and unique signature without staining. So that's important, that you don't have a staining in this, and it can give you so much details. And it does look at combination of both morphology and biomarker information. And here I'm showing it to you, these signals and how the HEG detects collagen fibers as in green, shown in green, and the other signal detects cells shown in red. So that has been revolutionary in the concept of histology in hepatology in general, in NASH and in NASH cirrhosis. And this is an example of studies that have been done by HistoIndex, looking at the Q fibrosis score. And this is just looking at fibrosis. We have not extended yet to the septal thickness and nodule. And these studies have been done on HBV and HCV viruses. And it shows you clearly the difference between cirrhosis and incomplete cirrhosis with thin septa, here shown in green. So the right and left look completely different. Also looking at the Q fibrosis in more detail, look at how many parameters you have here, 184 parameters. And I don't want to spell out of all of them, but look at, for instance, between the left and right. So on the left, you see non-cirrhotic sample, on the right, you see cirrhotic sample. And in the green is the zone 2 area that is more prominent in a non-cirrhotic sample, and it gets diminished in the cirrhotic sample. And you see here the nice color differences between the two samples, looking at different parameters in the portal tracts and periportal tracts. So this is the key fibrosis. And what the key fibrosis did in our study, we picked the top fibrosis parameters to build an index that correlates with HVBG. And here are two examples of correlations on one of the parameters that of the fibrosis correlated nicely with HVBG. Overall, this Q fibrosis correlated really well with HVBG. Moving on, we started looking beyond that and looking at the architect. And this is here when we focus on the septa, the length, the width, and many other parameters. And there were 18 parameters, and we looked at the septa number, the mean value in a certain area of the tissue, and we averaged that in that certain area of the tissue, which is very important. The septa occupies certain area and tells you how much you have septas there, which contributes to portal pressure. And this is the correlation between the septa parameters, the top 15 ones, and HVBG. And as you see here, left and right in the training and validation cohort, the correlation was very solid. Moving on to nodules. I mean, nodules should matter, right? We all know that. A bunch of large nodules is different from tiny few nodules. And here, look at this nice drawing that I think was done by HistoIndex or Dr. Zach Goodman, who was one of the influential people on this project, and look at the diameter of the nodule, the distance between septas, and many other features of the nodules to see how it correlates with HVBG. And here again, I'm showing you the third parameter correlation with HVBG, which is here the nodules. We took the top 15 nodule parameters and that were used to build correlation with HVBG index. And you see here the correlation is really nice. Now, the beauty of machine learning, you want to meld these in one index that here we call it the SNOOF or whatever you want to call it. S for septa, N for nodules, and on F for fibrosis. And the nice thing about this index or score that it takes in consideration very important architect features in cirrhosis and correlated with HVBG. And if you look at the bottom of that SNOOF score, in general, no matter what sample you look at the baseline or end of treatment, or you add one in or add one out, the correlation overall, if you combine the parameters, was much better than you have single parameter for septa, nodule, or fibrosis. So the combination of those histological features, septa and nodule fibers, perform that combination much better in correlation with HVBG. And here we said, we probably in the future validation studies and future studies, we can give away transjugular liver biopsy and use such a score. And one nice things about this score is that we found that the repeatability of the septa, nodules, and Q fibrosis are all above 95%. Indeed, the agreement between the septa nodule annotations that they came new to this game by pathologists versus by the algorithm was 91 to 95%. So there's a high degree of agreement with the pathology annotations, and in the future, those nodule and septa will be done in a systemic fashion in a machine learning algorithm. But let's look at the clinical data here. So at the bottom, you see HVBG, which again is invasive to predict varices, and the area of the curve was 0.75. If you look at the SNOOF score to predict clinically significant, the bottom to predict varices was 0.75. If you look at the SNOOF varices score, which was created to predict varices, it did better than HVBG in predicting varices. The area under the curve was 0.6. It was almost similar in the validation cohort, but overall performed much better than the traditional one in terms of sensitivity, specificity, PPV and MPV in predicting varices. The SNOOF score also predicted the clinically significant portal hypertension very nicely with area under the curve of more 0.85, and in the validation cohort was 0.75. And look at PPV and PPV values. It had very good numbers, and it's at least comparable to HVBG, the invasive one. Not even that, we looked at the changes of HVBG by 20%. So this is when the study, we looked at before and after, and if a patient had more than 20% change or not. And if that histological score could predict that, and sure it did with area under the curve of 0.89 and a validation cohort of 0.79, showing you the sensitivity of such a machine learning smart algorithm to predict changes via histology that correlates with HVBG changes, which are very important, especially that they correlate with outcomes. So key takeaway homes in this study that we went beyond the fibrosis concept in simplifying cirrhosis, and we really took in consideration the complexity of cirrhosis, adding septa and nodule to Q fibrosis. And that combining them in one score correlated nicely with HVBG, clinically significant portal hypertension, as well as presence of varices, which are very important parameters of liver outcomes, as well as surrogate markers for liver outcomes. And thus, we believe that using this HistoIndex machine algorithm, we can now measure important things in cirrhosis way beyond what we used to measure, and even correlating with important clinical outcomes such as varices without even doing invasive transjugular liver biopsy, rather just simple histology and analyzing it by this method. And eventually, we believe that this method, if applied in, for instance, in clinical trials for NASH cirrhosis, will be able to detect meaningful changes, small changes, to show off that your drug is moving the right direction, and detect tiny changes, which what we're looking for in the field of NASH cirrhosis, which unfortunately now is hard to reverse. I think that's my 20 minutes. I will pass the mic to my esteemed colleague, Professor Quentin Anstey, and I'll stop here, take questions later.
Thank you, Mazen, for your excellent presentation. Next, I'll welcome Professor Quentin Anstey with his presentation titled Development of Concordance Atlas for Stain-Free AI Digital Pathology in Balloon Hepatocyte Feature Recognition. The floor is all yours, Quentin.
Thanks very much indeed. And it's a great pleasure to be here to talk a little bit more about our work on identifying balloon cells. So these are my disclosures, which I'll just skip over. Really, what I'm going to focus on here is just one of the challenges we face with clinical trial design and drug development in non-alcoholic fatty liver disease. And that relates to the issues of inter-observer variation when interpreting liver biopsies. This isn't a new concept. It's something that's been recognized for some time. Here I'm showing you data from 2 papers written by Professor Kleiner, one from 2005, one from 2019, with the NASH CRN, where they published their kappa statistics for inter-observer variation. And you can see here, if we look at ballooning, the kappa statistics in both of these occasions was round about the 0.56, 0.57 zone, which is at best moderate inter-observer agreement. And of course, one has to remember these are expert histopathologists who meet regularly, look down a multi-headed microscope together. These are about as harmonized a group of individuals as you're going to get in terms of their histological interpretations. So the fact that over that 15-year period, there hasn't been a significant improvement in kappa values does make us think that there are limitations, and there must be something more to this than simply not practicing enough together. And this imperfect histological reference, of course, complicates the development of biomarkers and hampers drug discovery pipelines, as well as impeding clinical care. So it's really important we get to the bottom of this inter-observer variation. And in the clinical trial space, this is even more acute when we think about those balloon cells. You'll remember that to get into a trial for fatty liver disease, one tends to need to demonstrate a NAS score of at least 4, with at least 1 point from ballooning. So in other words, one has to be confident that balloon cells are present in the biopsy. And then one of the 2 key endpoints of clinical trials, that reversal of steatohepatitis with no worsening of fibrosis, specifically mandates resolution of NASH determined by a ballooning score of 0. So to get into the trial, we need to be confident ballooning is present, and to have a positive outcome in the trial, we need to be confident that ballooning has gone. And this is really something which we need to address, specifically when we consider those inter-observer differences. Although this particular study has some methodological areas which possibly need some further thought, it does highlight the implications of that for the conduct of clinical trials in terms of the level of agreement that might be observed, again, amongst expert pathologists. But one of the questions here is this variation in agreement a function of how the scoring system has been designed? Is this a problem due to how the score is allocated? Or is this something more fundamental related to how balloon cells are perceived by expert histopathologists? And the study I'm going to talk about today really seeks to address that key question about what is it that is leading to this degree of inter-observer variation, and how can we then best mitigate against it? And to deliver this study, I really want to acknowledge a phenomenal team of collaborators who came together, myself and Elizabeth Brunt, who co-led it from the academic side, Dean Tai from HistoIndex, and then, as you can see here, an absolute wealth of expert histopathologists and expert hepatologists, all coming together to address this key area. What was the study? What did it involve? Well, 9 expert hepatopathologists reviewed the liver biopsies, 10 digitized liver biopsies. And what we asked them to do was to circle each individual cell that they considered to be a balloon cell. And so this was done independently, 9 pathologists independently reviewing 10 digital histology images and independently selecting which individual cells they considered to be ballooned. We then waited 3 months. Those same images were represented to the pathologists, but this time in a different order. Some of the images had been rotated through 90 degrees, others had been mirrored to flip them over, and two questions were asked. The first question was, does this biopsy show NASH or non-NASH NAFL, in other words, steatosis or steatohepatitis? And secondly, please again recircle every single balloon cell that you have identified on the biopsy. And the big focus here, though, was primarily on inter-observer agreement. And here's an example of the sort of thing we can see. So a histological image here, and we can see that some cells have been circled by different pathologists to indicate the ones that they consider to be ballooned. And we can also see that there are some areas, for example, on the right of the slide, where many of the histopathologists agreed and they've all circled similar cells. And there are other areas where only one pathologist may have selected a given cell. What did we find? Well, over those 10 biopsies, as you can see here on this graph, there were substantial differences in the number of balloon cells that each pathologist perceived. Now, I should say at this point that the slides that they were presented with, we believed, based on a single pathologist's opinion, demonstrated a spectrum from simple steatosis to very active steatohepatitis. So it's certainly to be expected there are variations in the number of balloon cells which pathologists may identify. But what you can see here is, looking at the same section, there is substantial variation between pathologists in how many balloon cells they can be considered to be present, with slide 5, the one in the middle there, being the one where the majority thought that there were a lot of balloon cells present. And so the interclass correlation coefficient for consistency here was about 0.7, showing that by and large, the pathologists tended to agree if there were lots of balloon cells or very few balloon cells, that there was some moderate degree of consistency for burden of ballooning within there. But when we started to look at specifics of which cells the pathologists were actually identifying, and to look pairwise to see what degree of concordance there was, what we found was that between individual pairs of pathologists, the degree of inter-observer agreement varied anywhere from just 8% of overlap in terms of identifying the same cells up to 75%. And so, as you can see here on the heat map, there were certainly substantial areas where individual pathologists were not identifying the same cells. And so the kappa value for the presence or absence of ballooning as an absolute was 0.2. And if we took a threshold of at least 5 balloon cells to be identified to consider ballooning to be present, that kappa value increased slightly to 0.36, but nevertheless a low level of inter-observer agreement in terms of the specific cells that were being identified. And this posed a number of questions for us. We were keen to understand what this was about. And the first thing we looked for was, were there trends? And as you can see here, there clearly were. So each colour is a different pathologist looking at a different slide. And when the bar is going up from the x-axis, that indicates a pathologist who tends to see more balloon cells than the rest of their colleagues. And similarly, if it goes down from the x-axis, that implies pathologists who tended to see fewer balloon cells than their colleagues. And when they cross the dotted lines, that would be at least 1.5 standard deviations from the mean in terms of propensity to identify balloon cells. And so the takeaway messages here is that there are some pathologists who tend to consistently see more balloon cells, whereas others tend to be more conservative and see fewer balloon cells. An important point here, because it speaks to an outlook and the likelihood for identifying it. We wondered if this was simply related to cell size, and certainly there is some indication that cell size matters. So pathologists here shown in red who were willing to look at smaller cells and consider smaller cells to be ballooned tended to see more balloon cells, whereas pathologists such as the one shown in amber, who tended to only consider very large cells to be ballooned, tended to see fewer balloon cells. So size is one part of the story, but it's probably not the entire story, and there may be more to that. And in fact, when we considered over the 80,000 cells that were identified or looked at across these 10 images, there were about 1,180 cells that any one pathologist considered to be ballooned. But what I'm showing you here is the single cell out of those 80,000 that were examined, 1,100 that were considered to be ballooned. This is the single cell where all 9 pathologists agreed that ballooning was present. So this is, I think, as much as we can describe as the most classic image of a balloon cell that there is. We then went on and tried to convert that individual cell data into a semi-quantitative ballooning score to see if that would smooth the effects of the inter-observer differences. And here we can see that the kappa values for inter-observer agreement went up to about 0.3 with this approach of using a semi-quantitative scoring system. But if you look at certain of the slides, there was still substantial variation. A great example here would be if you look at slide 7, running vertically there, you can see that for slide 7, 2 pathologists considered there to be a very large number of balloon cells, enough to get a semi-quantitative score of 2, whereas another 4 of the expert pathologists considered there to be absolutely no balloon cells present. And it's not just that one image, the same sort of variation could be seen in several of the others. So there is definitely an issue here in terms of inter-observer variation and how it may be interpreted. We went on from that to consider whether the presence or absence of ballooning was truly the shibboleth, the indicator, if you like, of whether the pathologist considered a biopsy to be NASH or non-NASH. And here what I'm showing you is a heat map that demonstrates very nicely the more red a box is, the more balloon cells a particular pathologist considered on an image. The darker the blue, the fewer the balloon cells that given pathologist identified on a particular slide. And what you can see is that there were 6 cases, 6 slides where a pathologist made a NASH call despite having indicated that there was not a single balloon cell on that given slide. And so, interestingly, 2 things here. There was relatively little agreement on a NASH or no NASH diagnosis when pathologists were looking just at an H&E image. And also, there appeared to be something of a disconnect between burden of ballooning and the propensity to either diagnose NASH or non-NASH. There was only one slide where all 9 pathologists considered NASH to be present, which was that slide 5, which, if you remember, I demonstrated had very high levels of ballooning on it. And indeed, if we look at these 3 slides that I'm highlighting here, in each case, that semi-quantitative ballooning score was 0 for ballooning. But you can see in each case that the majority of the pathologists actually considered those biopsies to demonstrate steatohepatitis. So important implications here in terms of how we understand the histological definition of NASH, but also in terms of the inter-observer variation. And I think this is an important point that we really need to take away from this, that these data imply that there is very limited correlation between a determination of the absence of ballooning at either the absolute level or the less than 5 cell threshold level, and a pathologist's willingness to diagnose steatosis or steatohepatitis. And I think that probably speaks to the complexity of what is going on when a pathologist is integrating all the visual clues to identify a balloon cell. So this begs the question, can we make histology great again? Is there something more that we can be doing to standardize the reading and the interpretation. And a number of different approaches have been proposed to use artificial intelligence or computer-assisted measures in liver histology. So some rely on stained sections, but the one we're going to talk about here is an automated approach using unstained sections, which is, of course, two-photon excitation fluorescence microscopy, and then second harmonic generation, which looks for fibrosis. And these two techniques that HistoIndex has very much championed were what we leveraged next in our studies. So taking the individual balloon cells that we had identified and all the cells that had been circled, as I've mentioned, you can see here that if we consider the total number of cells that any one pathologist had identified, the top end there is 1,188 balloon cells, going down to 26 cells where at least 8 of the 9 pathologists had identified them, and going down to 1 cell where all 9 pathologists agreed it was ballooned, with a denominator of just over 80,000 hepatocytes being examined. Using the Q-ballooning 2, we were able to try training the algorithm using the inputs either from one pathologist or from 2 pathologists in agreement, 3, 4, 5, and so on, to see how the algorithm could be altered by using a defined training set which had a higher degree of pathologist concordance within it. And as you can see here, the more pathologists that are used to train the algorithm, the fewer the number of balloon cells that the algorithm identifies. But one can also see that the actual positive predictive value of the algorithm tends to increase, certainly up to a point. And we felt that the sweet point here was training the algorithm using the data set of cells for which at least 5 pathologists, a simple majority, tended to agree. And as you can see, that gives a positive predictive value of about 25%, as opposed to the human pathologists, with a false discovery rate of about 75%. Let me talk you through that a little bit more. The Q-ballooning 2 index then was built up using 7 parameters. 6 of those were purely balloon cell focus parameters, and 1 of them related to fibrosis and its association with balloon cells. And you can see here on this figure an example image. On the left-hand side, the cells that the pathologists had annotated, and then separately, independently, the blue-coloured cells are those cells that the Q-ballooning 2 algorithm was able to identify as being ballooned. And you can imagine in your mind, if you superimpose them, there's a relatively high level of overlap. What does this mean? Well, using a pathologist's concordance threshold of at least 5 pathologists as the reference standard, what this means is that Q-ballooning 2 trained with atlases containing at least 50 cells exhibited a sensitivity for ballooning which went from 11 to 41% and a specificity of greater than 99%. Although one must remember that specificity is of course a function of the fact that the majority of cells on any histological image are not ballooned. So I'd suggest focusing more on the sensitivity here than the specificity. And annotations based on at least 5 of 9 pathologists gave a 17% sensitivity with a positive predictive value of 25%, as I've already mentioned. Now that compares very favorably, in fact, to the performance of human pathologists in this study, where sensitivity ranged between 44 and 94% and positive predictive value from 13 to 53%. So what we're seeing here is that concordance data being used to train Q-ballooning 2 can help us train or tune, if you like, the sensitivity versus the specificity to either identify more cells but at the cost of a higher false discovery rate, or to detect fewer cells with a greater degree of certitude that these are indeed ballooned and would be judged to be so by the majority of pathologists. Thinking about it in a slightly different way, just to try to help us get a handle on what this means, earlier on, I showed you that the pairwise comparison between individual pairs of pathologists varied between 8% and 75%. And when we repeat that exercise, comparing Q ballooning 2 to pathologists A, B, C, D, and so on, again as a pairwise comparison, we found that the agreement here ranged between 19 and 42%. In other words, very much within the same range of performance and inter-observer agreement that one would see between pairs of expert human pathologists. But of course, there is no intra-observer variation with a computerized algorithm. It does the same thing every day. The final thing I just want to highlight here is the question of whether these sort of algorithms can be sensitive to change. In other words, able to detect change when there is a histological response. Now, this was a small adjunct study, and really we included it in our paper as a proof of principle. But what we did was we took paired biopsies from a clinical trial, and we divided those biopsies into those where the central trial pathologist had considered that there were a reduced number of balloon cells, and those where the central trial pathologist had considered there was no reduction in the number of balloon cells. And what the graph shows you is that there was actually relatively good concordance here. So where a central pathologist would consider that there was a reduction in the number of balloon cells, the algorithm similarly detected a reduction in balloon cells. And for the cases where that was not felt by the central pathologist, again, the algorithm tended to see, in fact, a small increase in the number of balloon cells. Now, that part is very much proof of principle and will need further validation, but is nevertheless very encouraging in terms of the ability to use these AI-based approaches in future trials. So how can I conclude? Well, despite the inherent flaws in semi-quantitative scores such as NAS or SAF, histological assessment remains a cornerstone of clinical trial conduct. Hepatocyte ballooning identification is necessary for trial recruitment and also as an efficacy endpoint. We report substantial inter-observer variation in balloon cell identification by expert hepatopathologists and show limited correlation between a determination of the absence of ballooning and discrimination of NASH or non-NASH NAFLD. Our data suggest that ballooning is probably too subjective for an absolute presence or absence call to be used as a clinical trial endpoint. And we need to potentially move to a more quantitative, a more nuanced approach there. We also demonstrate, however, that a concordance atlas leveraging this large data set of tightly defined balloon cells may be used to help train artificial intelligence-based algorithms in order to optimize their performance. And I'd hope that with further development and validation, the use of AI-assisted technologies may well help us to standardize these crucial histopathological assessments and provide a measure that more fully captures changes in disease severity. And really, I just want to reinforce one final point here. This isn't an issue about the fact that human pathology is not adequate to the task. It is the fact that we are requiring of human pathology something that really these semi-quantitative scores were not designed for. And so it's really a case that we're asking a human to do a machine's job, and maybe we need to be thinking about that slightly differently in the clinical trial setting. Once again, I'd really like to acknowledge a phenomenal group of collaborators and to point out that this is the first step in some ongoing work and that there'll be more from this research team going forward, and to thank you all very much indeed for your attention.
Thank you very much, Quentin, for taking us through the findings of this ballooning study. With the end of our presentations comes the much-awaited Q&A segment. It's great to see that we have a variety of questions here coming in, and we will do our best to answer them in the next 15 minutes. So I'll hand this over now to my colleague and moderator, Anthony Lai, the Head of Clinical Affairs, to facilitate our Q&A segment. Over to you, Anthony.
Thank you, Cynthia. A very good day to all of you. And once again, thank you, Dr. Mazen Noureddin and Professor Quentin Anstey, for the wonderful presentation that you have shared with us earlier. We do have a number of questions from the audience, so let's dive in right now to the first question. So the first question is for Mazen. How can the SNOF score try to build more granularity in cirrhotic trials compared to a single score fibrosis scoring? And will it better tease apart decompensated cirrhosis patients for a more targeted mechanism of action or trial study?
Yeah, I mean, that's actually the goal why this score was designed, is to include more features beyond just the one line of fibrosis and how it's moving, and include important architect, including the nodules as well as the septa. I don't want to repeat the presentation, but when I was reading this literature from 2006, the goal was then to subclassify even child A cirrhosis into multiple pockets that are different in severity and that can affect outcome differently. I'll give you an example from the NASH literature. There was a paper in JHAP. The first author was Zubair Younasi, which is the Gilead data that showed that cryptogenic cirrhotics they had more severe outcomes and they had more collagen area in general. So this work eventually, we hope to take, we need more studies to subclassify patients even within the child A cirrhosis into different severity, maybe to stratify by them when we do cirrhotic trials, or maybe just to do a trial completely designed on one of these subclassifications and see if the MOA will target that subclassification well and cure it better. You usually try to think that you want to start with a lesser degree of disease for your molecular to work. But again, it depends on the molecular. So we need more steps in general, but this is the most important first step, including this architect, and then more clinical data needs to come. One example, I'm sorry, I don't want to take too long. One example can be using these scores that patients with or without varices, you can subclassify by that they will get to clinically significant portal hypertension or not, and subclassify them into these groups and either re-stratify or do trial completely on those kind of more precision histology method.
Yeah, thank you for that. So just to follow up on to the to your answer, there is the question that came in. The use of the SNOF seems to be promising. Will you then advocate the use of the SNOF model for all NASH cirrhotic clinical trial, and why?
Oh yes, that's the answer. No, we have to do it in a systemic approach. Eventually, I would love to see machine learning and AI histology in all clinical trials, like the ballooning that was talked about today, in cirrhotic trials, but we always have to validate it, make sure it's validated in other studies, and start including in more and more, and look at also change in outcomes based on it. So yeah, eventually I would love to see it in all, but with the steps that the regulators kind of outlined it for us.
Great, thank you. So the next question is for Quentin. How can AI, as you mentioned in the presentation, 99% reproducibility, try to improve reproducibility rate for balloon annotation in a phase 2 or 3 clinical trial, and even up to the post-drug approval stage where there are hundreds or even thousands of patients involved?
Yeah, so well, let's take the clinical trial part first of all. And I think what we need to consider here is we need to separate out the concept of making a diagnosis. This is fatty liver disease, this is not autoimmune hepatitis, from the concept of assessing the minutiae of that disease and its individual components. And I think in the clinical trial space, this is where AI really comes in. In my day-to-day practice, I don't ask my pathologist to tell me, are there 1, 10, 20, 30 balloon cells on a biopsy? That's not what I'm interested in. But in a clinical trial, I'm looking for a degree of consistency that can be readily applied again and again to confirm that an individual had a minimum threshold to enter a study, and that same minimal threshold has been crossed in terms of a treatment response or not at the end of a study. And I think that's where AI comes in, because it provides a quantitative and reproducible measure that can then be used to standardize patient enrollment, not subject to inter or intra-observer variation at the start of the study, and also standardize that assessment of therapeutic response later on. And I think that would be a very helpful thing in removing some of that variation that we know impacts on clinical trials.
Great, thank you. So I do have another question from Abhishek Roy. So this question is, given the cost of SHG imaging, how do you see this technology being used in clinical practice? So the question can go to both Mazen and Quentin.
Yeah, well, I think that one may be one actually for you, Anthony, in terms of the cost of it. What I think Mazen and I can probably comment on is the performance of it and what it offers. The question about how then that can be scaled up is a slightly different thing, and I think that would probably be outside the remit of a jobbing hepatologist such as myself or Mazen.
Yeah, I totally agree with Quentin, but I also want to add that look at the MOAs that have failed so far, and some sure did not have a viable mechanism that could have continued. But we have also seen data that the inter and intra-observer variabilities likely affected many trials. And look at the amount of the money that getting wasted by having trials failing because of that. And the point is we definitely need to improve on that as a priority and as a cost. So I'll leave the details, as Quentin said, to HistoIndex, but it's totally justifiable to go that route for one, patient's sake, and two, for the field's sake.
Yeah,
great. So Quentin, I have another question over here. This is from Lucas. Given all the variability in identifying balloon cells, do you think that machine learning algorithm approach can be improved by identifying other features of steatohepatitis, NASH? And for example, why did you focus on 6 balloon cell parameters and only 1 fibrotic parameters? Was this just to have a starting point, or were other parameters less obvious?
Right. No, this is a brilliant question, and possibly I didn't explain myself well enough there. We actually started off with almost 100 different parameters, which were reflective of ballooning, and then we used a reductive statistical model to try to condense that down to a small number of parameters that gave us the greatest degree of accuracy. So I brushed over that, but that was the statistical approach there, as we built, if you like, a regression model which associated with ballooning. So no, we started off with many, many more than that. We actually looked at a whole range of features, those specifically related to ballooning. I've mentioned also some issues related to fibrosis, because of course, the reason we all are interested in ballooning is because we believe it is relevant for fibrosis, and then we condensed it down into the Q-ballooning 2 index, which contained those specific features. And if you're interested in what they actually are, they're all listed in the paper. So it's all there to actually have a look at.
Mm-hmm. Okay, thank you for that. And we have come to our last question. We have time for one more last question. So this is an interesting question, and I'd like to ask both of our speaker and highly esteemed KOL in NASH to share their thoughts on it. We see the value of this technology involving both SHG and machine learning. Do you think that AI assistive tools such as SHG image and fully quantitative assessment from HistoIndex can aid the pathologists during screening to reduce screen failure rate and evaluate end of treatment drug efficacy? Yep.
Short
Quentin,
answer,
would you go
yes.
first? Yep. Mm-hmm.
Yeah, no, it's a very short answer. Yes.
Indeed.
I think we need to think about how that works. There are a number of different models. One is that, one runs the algorithm then presents the cells it considers ballooned to the pathologist, and the pathologist then, if you like, validates that response and uses it as an assisted thing. And to be honest, that's something we're already beginning to see in radiology in routine practice, where AI is helping us to spot, pulmonary nodules and so on and so forth. So it's not a million miles away from that. So I think there is certainly scope to help with that. At the other end of the spectrum is the complete quantification. So the pathologist examines a biopsy, confirms that it is the right disease, not autoimmune hepatitis, not viral hepatitis, and then the biopsy is passed over to the AI technology to give a quantitative readout. And that's another way that this could be used. I think there's a lot of discussion needed here, particularly with the regulators in terms of what they're ready to accept and what level of evidence we need to make those moves. But undoubtedly, we are at the time where we need to start thinking about this, because as Mazen has indicated, we are probably at the risk of losing efficacious drugs, not because they don't work, but because we are not accurately measuring how they work.
And I totally agree with Quentin. And if I want to pick one, it's probably my first choice. And I want to give the example of the self-driving Tesla. I will get into it if there's a driver that will eventually control and say the final say, which is our esteemed pathologists that we all admire them. But why not do it in a high technology systemic way, while I have like my confident driver that the machine is doing the same thing over and over. He's just validating it. So that's how I look at it.
Thank you, Mazen, for that. Yeah, and that was a very nice analogy that you used, using Tesla as a fully automated car, having a driver inside as well. Okay, thank you, Mazen and Quentin, for the excellent presentation, an important session which I believe will add great value to the NASH community, especially in the context of NASH clinical trial. We've come to the end of our Q&A segment. You may be keen to read up on the Q-ballooning paper that Quentin has just talked about, and I would like to share with you where you can get a hold of this paper. The balloon paper titled Complexity of Balloon Hepatocyte Feature Recognition, Defining a Training Atlas for Artificial Intelligence-Based Imaging in NAFLD is published in the Journal of Hepatology, and it is available as a free access for download. You can also find the ballooning paper presentation during AASLD and the ballooning publication in Journal of Hepatology on our HistoIndex website for easy access. The poster presentation on development of machine learning histological scores that correlate with portal pressures and development of varices in NASH patients with cirrhosis that was shared during last year's AASLD by lead author Dr. Noureddin is available for download on our HistoIndex website. Besides this, I'm very pleased to share with you the latest episode on the Surfing the NASH Tsunami podcast, where KOLs in NASH such as Dr. Stephen Harrison and Professor Johan Schattenberg share their thoughts and discuss at length with Dr. Mazen Noureddin and Professor Quentin Anstey about the two topics they have just presented. In addition, we have our Chief Scientific Officer, Dr. Dean Tai, who spoke about the informed application of AI Digital Pathology in improved balloon hepatocyte identification, the analysis in advanced fibrosis and cirrhosis, and what more can be done for the NASH community. So to conclude, HistoIndex is in about 40% of all phase 2 and phase 3 NASH clinical trials. And please feel free to reach out to us at info@histoindex.com to see how we could partner with you and add value to your clinical trial. With that, I would like to thank both our panelists again, our participant, and Cynthia for being such a great host. Thank you for listening, and I'll hand the mic back to Cynthia.
Thank you too, Anthony, for being such a wonderful moderator. Now, we've come to the end of our webinar, and I'd like to say some thank you messages. First of all, I'd like to thank our co-organizer, Global Engage, for their excellent support. We'd also like to extend our great thanks and appreciation to our presenters, Dr. Mazen Noureddin, Founding Director of the Fatty Liver Program at Cedars-Sinai Medical Center, Los Angeles, California, and Professor Quentin Anstey, Chair of Experimental Hepatology and Consultant Hepatologist from the Faculty of Medical Sciences at the Newcastle University in UK. Thank you so much, presenters. And to our guests, it was a pleasure being with you today and answering your questions. Now, this webinar will be made available on demand in the next few days, and you will definitely be informed via email. So please look out for our message. Now, if you have more questions for us, please drop us an email at info@histoindex.com. Also, please stay tuned to our website at www.histoindex.com, or you can stay tuned to our LinkedIn page for further updates. Thanks again for joining us today, and we will see you next time with more content and data on stain-free AI digital pathology. Have a nice day. Goodbye and good night.
Thank you.
Thank you.
Bye-bye.
Bye.
Goodbye. Thank you.